So welcome to all of you, and today we are going to discuss the instrumentation of IR spectrometer. Okay, so you can see this one instrumentation. First, we will discuss the main parts of IR spectrometer. Okay, so the main parts of IR spectrometer will be the radiation source. First one, second one is sample cell or sampling of substances. Third is monochromator detector, and finally recorder. Okay, so these parts we will discuss step by step. Okay, so first part is your infrared sources. So what is the infrared sources used in IR spectrometer? In IR instruments require a source of radiant energy which emits IR radiation, which must be, there are three qualities of IR radiation: that it should be sufficient in an intensity. Okay, second one is it should be continuous, and third one is stable. Okay, so these are the some qualities should be IR radiations, and for IR radiation different types of sources it is used. Okay, so sources of IR radiations are as follows. First one is globar. Okay, so globar chemically made up of silicon carbide. Okay, and silicon carbide sustain less temperature, and you can heat it 30 degrees centigrade. Okay, so after heating it produces radiant energy from 1 to 40 micron. Okay, so it is the glow bar. Okay, so glow bar made up of rod of silicon carbides, and you can heat it into 13 degrees celsius for producing the radiant energy from 1 to 14 microns. Okay, next one is nuts glower. Okay, nuts glower sustain higher energy as compared to glow bar. Okay, and it is as made up of zirconium and yttrium. Okay, it is F block elements, and you can heat 1500 degrees centigrades, and it emits radiation between zero point 0.4 to 20 micron. Okay, so you can differentiate between nuts glower and glow bar. Okay, it's chemically is different and sustaining different temperature. Okay, and producing the different types of radiations in microns. Okay, so next one, second one is the sample cell. Okay, so for gas samples the spectrum of a gas can be obtained permitting the sample to expand into an evacuated cell, also called cuvette. Okay, so this is your cuvette, and in this you will put the sample, gaseous sample. Okay, and it is closed. Okay, and after passing IR radiations, okay, it further proceed for IR spectrometer. Okay, for giving the final spectrum. Okay, so for solution sample, infrared solution cell consists of two windows, a pressed salt shield sample that are liquid at room temperature and usually analyzed in pure form or in solution form. Okay, so either you can put the pure form of sample, or you can form the solution, and for making solution you will use the carbon tetrachloride or CH2, means carbon disulfide. Okay, so there are two you have to means either you can take the pure sample in KBR, mix up in KBR and form the pellets and analyze that. Okay, or you can also form the sample, means sample in CCl4 and carbon CS2. Okay, so it is the medium for mixing the sample and forming the solution. Okay, so it is the solution sample. Okay, next we'll talk about the solid sample. If you have solid sample, then how you can make the pellets. Okay, so for solid sample, first of all the solid particle will be reduced to small particles. Okay, and you will make less than 2 micron. Okay, and after that you will examine. Okay, into means first of all you will form the thin paste, means you will grind the solids into small particles, and it will be less than 2 micron. Okay, after that you can examine. Okay, and you can, after that what happen here, you can use KBR. Okay, either you will use KBR, and in KBR what you will mix the sample. Okay, so sample and KBR will be mixed, and you will form the pellets. Pellets is spherical type, and please you should examine that your KBR pellets will be uniformly, okay, and it should be thin, and there will be no crack will be present, okay, either, okay. So IR radiation will passes through these pellets will be very clear way, okay. So all the samples will absorb IR radiation uniformly, okay. So here actually I have mentioned that solid reduced as small particles first of all can be examined as a thin paste or mold. The mold is formed by grinding a 2 to 5 milligram of the sample in the pressure of 1 or 2 drops of hydrocarbon oil, okay. Or you can say nozzle oil is also used. The resulting mold is then examined as a film between plates or plates, okay. So it is the method for used in the for solid sample, okay. Next we will talk about another technique is to ground a milligram of less than milligram with 100 milligram potassium bromide. The mixture is then pressed and evaluate dye to produce the transparent discs, okay. So actually I have discussed before, I have discussing before, okay. So in this what happened, I have discussed in this. Then first of all you will use KBR and sample your sample, okay, and grind it very finely, and after that you will make the means you will press that mixer. So that's why you will for you can obtain the uh, you pellets, okay, okay. So it is the actual instruments where pellets you can form here, okay. So next one is radiation detector, okay. So infrared detector is a detector that reacts to infrared radiation, okay. So infrared detector is a detector that reacts to in IR radiations, and it's simply a transducer of radiance energy, okay. So आपका जो भी if you are going to use detector, then it is able to react to IR radiations, okay. So it is the qualities of detector, okay, and it is simply a transducer of radiance energy, okay. So radiant energy means it absorbs the infrared radiations, okay. So detector is two types, either it will thermal or non-thermal. Okay, so two types of detector is used, either it may be thermal or non-thermal, okay. So what is the difference between thermal and non-thermal detector? In thermal detector can be used over a wide range of wavelength, and they operate at room temperature, okay. So it is used for the wide range of wavelength and operate at room temperature. Disadvantage is slow response time and lower sensitivity related to other types of detector, okay. So it is the drawback of this detector, and it shows the slow response time and lower sensitivity related to other types of detector, okay. So type of thermal detector, if you talk about the types of thermal detector, so it is bolometer, thermocouple and thermopile, pyroelectric detector, and Gole cell, okay. So it is the types of thermal detector, okay. So what is the bolometer? So bolometer is derived from a Greek word bolometron, okay. Bolo means for some time thrown, and metron is measure, okay. So construction, a bolometer consists of absorptive elements such as thin layer of metals. Most bolometer use semiconductor or superconductor absorptive elements rather than metals, okay. So it is the composition of bolometer, okay. Working thin layer of metal connected to a reservoir, okay, and any radiation on absorptive element raises its temperature above that of reservoir. The temperature changes can be measured directly with attached thermometer, okay. So it is the process of working of bolometers, okay. Next we will talk about the thermocouple and thermopile, okay. So thermocouple consists of pair of junction, okay, of different metals. For example, two pieces of bismuth fused to either end of a piece of antimony, okay. So thermopile temperature changes creates the potential difference, okay. So okay, so this is the T1 and this is the T2. So when temperature changes occur, these thermocouple in thermocouple you can see the potential difference, okay. So thermocouple detector are voltage generating devices which can be thrown off as mounted miniature areas of thermocouple junction, okay. Pyroelectric detector is single, you can say the construction of this single crystalline wafer of pyroelectric material such as triglycerine sulfate, okay. So it is the pyroelectric material triglycerine sulfate, okay. Pyroelectric infrared detector converts the changes in incoming infrared to electric signals. Okay. So next will talk about the processing of pyroelectric detector. So below Curie's temperature, pyroelectric material exhibits electrical polarization. Okay, temperature is altered, the polarization changes and observed as an electrical signal. Okay, next we will talk about the Gole cells. So construction is a small metal cylinder. It is the small metal cylinder and having flexible silver diaphragm. The whole chamber is filled with xenon gas. Okay, and so it is the, you can see this one, it is not very important, but you should learn this one. Okay, so non-thermal detector work as, first of all, thin film overlaps over non-conducting surface, absorption of IR radiation and resulting the non-conducting valence electron to higher energy conducting state, and electrical resistance decreases, so that's why voltage drops. Okay, so for next one is photovoltaic detector. Okay, so you can see infrared radiation, photovoltaic detector generates a small voltage, detect as a signal. Okay, so last you can see the signal will be appeared. Okay, so IR absorption spectrometer is two types: single beam spectrophotometer and double beam spectrophotometer. It is very important. So a single beam of light which can passes through one solution at a time is called single beam spectrophotometer, while a single beam of light is split into two separate beam. One passes through the sample and another passes to the reference. So that's why it is called double beam spectrometer. Okay. So single beam IR spectrophotometer is radiance source. It is a radiance source. It is the sample cell, monochromator and detector. Okay. So detector finally creates the signals. Okay. Similarly, you can see single beam IR absorption spectrophotometer. Uh, okay, so you can see this one. Double beam IR spectrophotometer. You can see here two types of. This is the one beam, and second one is the this one. Okay, so that's why it is called double beam absorption spectrometer. And okay, you can see this one, this part. So by which it acts. Okay, so finally. Uh, it is connected to you can see radiation source passes the sample and references, and after that attenuator is attached to the monochromator and detector, then you can see this one. Okay, double beam, it is not very important, so that's why we are neglecting that. So next one is Fourier transform spectrometer. It is very important. So all frequencies are examined simultaneously in Fourier transform infrared spectroscopy. Okay, so all the frequencies simultaneously will be examined through Fourier transform spectrophotometer. Okay, so application, we will talk about the application of IR absorption spectroscopy. So since different molecule with different combination of item produce their unique spectra, infrared spectroscopy can be used in to qualitatively identify substances. Okay, so what is the qualitative analysis? You can see one. Uh, this is the group frequency region and this is the fingerprint region. Okay, so in fingerprint region there are lots of signal will be appeared. Okay, or band will be appeared. Okay, so in this group frequency region consisting of the absorption band of the functional group. Okay, these functional group majorly appears in 1300 to 400 centimeter inwards. Okay, and you can see if a wavelength 2.5 to 8. Okay, and fingerprint region is called because no other chemical species will have similar IR spectrum. Single bonds give their absorption band in the region frequency 1300 to 650 centimeter inwards, wavelength 8 to 15.4. Okay, so you can see this is the region of group frequency region and this is the called fingerprint region. Okay, application of IR spectroscopy to organic molecule. Organic groups differ from one another both in strength of the bond and the masses of the atom. Okay, so different molecules having the different masses and their attachment of bond. Okay, so you can see the alcohol and amines will appear in 13 to 400 centimeter inwards. Complex interaction will appear in 909 to 1300 centimeter inwards, while benzene rings appear in 650 to 909. Okay, so it is very important frequencies for different functional groups. Okay, So studying progress of reactions, observing rate of disappearance of characteristic absorption band in reactions, reactants, rate of increase absorption band in products of a particular products. Okay, so OS peak will appear in 3650 to 3600. Okay, while CO will appear 1680 to 1760. Okay, so hope you will like our presentation. Thank you. And in the end of this lecture, we will ask to you that please neglect these detectors. Okay, because I did not explain very clear way. This one means this like this single beam spectrometer like that. Okay, so it is not very important for you now. These ones. Okay, so you will only concentrate the IR spectrometer means what means which type of parts is used in the IR spectrometers. Okay, so finally thank you for all for watching this video. Okay.